Hi, my name is deoxyribonucleic acid, but everyone just calls me DNA, and this is my Draw My Life. I was born in this place called the nucleus, where I spend my entire life. My job inside of the cell is to share all of the blueprints or genetic information to make pretty much everything inside the cell. It's kind of an important job. I'm constructed of 2 strands, making a double helix. Some people tell me I look just like a ladder. My basic unit of construction is called a nucleotide. These little guys are my monomers, made up of 3 things: a pentose sugar, a phosphate group, and a nitrogenous base. My strands are anti-parallel, meaning that the nucleotides run opposite of each other. The pentose has 5 carbons. One strand runs from the 3' carbon to the 5' carbon, and the other strand runs oppositely from the 5' carbon to the 3' carbon. The 4 nitrogen bases are adenine, thymine, cytosine, and guanine. A bonds with T and C bonds with G. This is because single-ringed pyrimidines have to bond with double-ringed purines. This combination provides the perfect distance between the 2 strands. This is also why A and T are always in equal amounts, as well as G and C. Let me explain how I'm made. The process is called DNA replication. It all starts with a parental DNA. My mom's strands were replicated to create me, the daughter DNA. First, an enzyme called helicase comes to unzip the genes. Where the nitrogenous bases are separated is the origin of replication, which forms a replication bubble. The place where the parental DNA is unzipping at each end of the bubble is called a replication fork. Next, single-stranded binding proteins come and hold onto the strands to make sure that they stay open for replication. Plus, topoisomerase helps to relieve the strain on both sides of the helix. He's kind of like my masseuse when I'm all tensed up. Now this is where it gets really cool. Now that the nitrogenous bases are exposed, RNA primers can be laid down by the enzyme primase to start replication of each strand. The enzyme DNA polymerase adds complementary bases to each parental strand. This enzyme works smoothly on the strand that runs 3 to 5, since DNA polymerase can only run in this direction, producing a new 5' to 3' complementary strand. The other, which runs in the 5' to 3' direction, makes this next part trickier. RNA primase has to lay down multiple primers in order for polymerase to make a 3' to 5' strand. These form smaller fragments of DNA called Okazaki fragments. Since this process takes longer, it is referred to as the lagging strand, while the other is the leading strand. Finally, to bind the backbone, DNA polymerase 1 removes the RNA primers, while DNA polymerase 3 fills in the missing DNA sequence. DNA ligase then seals the backbone together. My enzymes are really smart and precise because they proofread me during my replication and repair any damage. There's an error rate of only 1 per 10 billion nucleotides. We've got this down to a science. Sometimes replication can lead to mutations, or errors in DNA sequence. After I was being replicated, I had to be repaired because I had an error in my sequence. A nucleotide excision repair was performed. An enzyme, exonuclease, found and cut the damaged segment, and the gap was filled with the help of the enzyme DNA polymerase and enzyme ligase. Thank goodness they fixed it. The ends of my molecule are different than the rest of me. They consist of multiple repetitions of one short nucleotide sequence. This is called a telomere. Although my telomeres don't hold genes, they are still important because they keep my genes from eroding after multiple replications. This means that I can pass on all of my genes to my daughter DNA. The enzyme telomerase adds onto the sequence to the ends of DNA, adding protection. I know it sounds like I'm making all of this up, but I swear I'm not. This replication happens all the time, and it's how I was made. Thanks for listening to my story. Bye!